Dear Neet Cognition Team Doctors, welcome to day 6 of Neet Neural Log series. Today we are going to see about the important organelles and their functions. But before that, shall we have a recap about yesterday's class? Then how was your day today? Did you all hydrate? Had some snacks? Fuel your brain? Then you can start studying, because that is very important. Yesterday what we saw is block 1. Under that we saw vacuole. The single membrane of vacuole is called as Tonoplast. Plant vacuole occupies 90% of the plant cell volume. Example of food vacuole is Protista. For contractile vacuole it is Amoeba. What is the function of contractile vacuole? Osmoregulation and excretion. Then we did semi-autonomous organelles, that is mitochondria and chloroplast. The reason for them to be semi-autonomous is their own circular DNA and 70S ribosomes. They are double membrane organelles. These are the similarities. Both have relationship to endosymbiotic origin. Then the 70S ribosomes shows prokaryotic similarity. That is why they have included in endosymbiotic origin. And yesterday I forgot to mention one point here. Both divide by binary fission. Then how many of you did repetition? Practiced repetition. Yesterday I mentioned on mental hack. How many of you started practicing that repetition? Today what shall we see? One important thing, okay? One important mental hack, that is about procrastination. Okay, first we'll start with the Neet Neural Log series day 6. First with the organelles, okay? The first organelle we are going to see is the nucleolus, which is the genetic command center of the cell. The major function, it has another function, that is it carries the genetic blueprint, but I have mentioned here as few of the major functions, okay, for you to remember, because we are writing only code words, right? So what is the major function of nucleolus? It is the site for rRNA synthesis. R is the stands for ribosomal RNA synthesis. What are the other RNA? You know, you have mRNA, tRNA and rRNA. Okay, do you know which lesson it comes? If you know it, type the answer in the comment section. What is the primary product which comes out of this rRNA synthesis? See, R stands for ribosome site also. It prepares ribosome subunits. The primary product or the action is preparation of ribosome subunits. What are these ribosome subunits? You have ribosome organelle inside the cell, isn't it? That ribosomes are synthesized by the nucleolus. Okay, so shall we start writing it 3 times? Nucleolus, which occupies the nucleus. See, I am talking about nucleus. It is not nucleolus, okay? It's not nucleus. There is a difference. We saw already nucleus surrounded by nuclear membrane. Inside that, the matrix you call it as nucleoplasm. Then what else? We saw chromatin, fibers. Then we read about that spherical bodies, that is called as nucleolus. Here the spelling changes. See, in the exam tension, you will be in a panic to see these spelling, okay? This is nucleus. Here see, it is nucleolus. So here I am talking about nucleolus, the spherical bodies inside the nucleus. I'll draw it here. This is nucleus. Here spherical bodies inside this is the nucleolus. Okay, got it? So this nucleus synthesizes nucleolus. Sorry, this nucleolus synthesizes rRNA. Now shall we start writing nucleolus? What is the function? rRNA synthesis. Function of nucleolus, rRNA synthesis. I am very happy to welcome the students who have joined here. It makes me feel really delighted. Welcome, welcome to Neet Neural Log series students. We are going to do it together. So these rRNA synthesis are preparing the ribosome subunits, which is the ribosome of the cell, that is the ribosome organelle. So nucleolus is the site of rRNA synthesis, which prepares ribosome subunits. Then the third time, nucleolus is the organelle, site of rRNA synthesis. The primary product out of this is ribosome subunits, which are the ribosomes of the cell. Now, the second organelle is Golgi apparatus. What do you know about Golgi apparatus? If you know the answer, type it in the comment section quickly. Okay, they are involved in packaging and modification of materials. Can you tell me one example, students? Packaging and modification of materials. What does it mean? See, for example, insulin, in synthesis of insulin hormone. Okay, it follows the, yesterday we saw about endomembrane system. Okay, endomembrane system, right? So from endoplasmic reticulum, it reaches Golgi bodies. This endoplasmic reticulum is a raw manufacturer. Okay, raw manufacturer. But there needs to be some modification for it to act as an insulin. So that modification is carried out by means of Golgi apparatus. See, what is the product? Glycoproteins and glycolipids. That is, the raw proteins from endoplasmic reticulum are glycosylated. Addition of group to proteins and addition of group to lipids. We'll saw it in detail. Okay, so shall we start writing now? Glycoproteins and glycolipids, that is addition of the group to the proteins and lipids, takes place where? In Golgi apparatus. Golgi apparatus helps in packaging and modification of materials, that is the raw materials from the endoplasmic reticulum. Endomembrane system, you have only few lines in the NCERT book, but it explains a lot. Okay, we'll do it with a model in the upcoming videos. It produces what? Glycoproteins and glycolipids. Sorry, I forgot to add the space here. Golgi apparatus helps in packaging and modification of materials, that is what? Addition of group to the proteins and lipids, so that it completely transforms into a hormone. Okay, or whatever the product it has destined to be. Okay, next we have ribosomes. Ribosomes, these are the organelles, very important organelles who supplies proteins to all the functions of the cell. Okay, functions of the cell. If there is a need for protein inside the cell, the ribosomes has to supply all the proteins. Now we saw where do the ribosomes come from? It comes from nucleolus. Now you understood this nucleus is a very important organelle for this cell. So what is the function of ribosomes? Protein synthesis. Protein synthesis. We too need protein, right? So Protein is very necessary for what? Can you mention one use? Okay, they are very necessary for muscles. Okay, these proteins are nothing but polypeptide chains. Polypeptide chains. Polypeptide chains. They are polypeptide chains. Next is the rough endoplasmic reticulum and smooth endoplasmic reticulum. Why do they give the name rough and here smooth? Because ribosomes attached to endoplasmic reticulum are called as rough endoplasmic reticulum. If there is no ribosome, that is absence of ribosomes, means it is a smooth endoplasmic reticulum. So remember, R stands for ribosomes, which means rough. If there are no ribosomes, it means smooth. Now, what is the function of rough endoplasmic reticulum? Protein synthesis and secretion. Can you name one secretory protein? The hormone insulin. Okay, you call it as hormone. As I said, like after all the modification, packaging, everything over, the hormone is released into the blood. But you call it as a secretory protein. Basically, it is a secretory protein. So, rough endoplasmic reticulum, or the function of rough endoplasmic reticulum is protein synthesis and secretion. So, we'll write it protein synthesis and secretion. Students, for those who want to only live class as a teaching session, we will do. Today I've got the idea to split it into 2 live sessions. That is, one I'll explain and I'll finish off. The second one is the idea of this series. The main goal is to keep it writing, because when you write, it gets logged in your long-term memory. This is one of the secret of the toppers. Okay, I sit down and write with you because you should know, like I am teaching for you. As a teacher, I should also practice this. There is no shortcut for success. If you want something badly, you have to do whatever it needs to be. Okay, there is nothing wrong in writing it 3 times than wasting a 365 days as a dropper. Okay, I think you got the point. Now continuing it, protein synthesis plus secretion. Protein synthesis. Rough endoplasmic reticulum is involved in protein synthesis and secretion. So the final product, what you get is secretory proteins. The final product is secretory proteins. What is the final product? Secretory proteins. The final product is secretory proteins. Now, what is the function of smooth endoplasmic reticulum? The function is lipid or steroid, that is hormone synthesis. You have steroid hormones, right? So, smooth endoplasmic reticulum helps in lipid or steroid, that is hormone synthesis. Then what is the main action? Lipids plus detoxification. What is the meaning of detoxification? If you know the answer, type it in the comment section quickly. Detoxification is related to drugs. Okay. So if you know the answer of meaning of detoxification, type it in the comment section. What is the function of smooth endoplasmic reticulum? Is to produce lipid or steroid hormone, that is lipid or steroid synthesis. Lipids plus detoxification. The final organelle is the lysosome. It helps in intracellular digestion. That is, it acts as a stomach for the cell. Stomach for the cell. What is the action it does? It digests the waste and biomolecule. That is biomolecule breakdown. Lysosomes contain hydrolytic enzymes. That is the function. How should you call the function? It is called as intracellular digestion. What it digests? That is, it digests all the waste and biomolecules. Okay, biomolecule breakdown. Now today I am going to share a mental hack, students. I'll be finishing off today a bit quickly because I'm feeling a hangover of my drug withdrawal, but still I want to show up today. I don't want to break the consistency. Even if it is 15 minutes, I plan to do it. So today I'm going to talk about procrastination. The mental hack is about procrastination. You know, it is about postponing things. Okay, postponing whatever you have planned. Now I've seen a lot of videos about procrastination and laziness, particularly laziness. So students, in your mindset, what you think is you all feel that you are procrastinating, you all have become lazy, but actually the fact is not this. Okay, procrastination is not about time management. It's not only about time management. It is actually a emotional regulation. You need to take it as an emotional regulation thing. Okay, don't focus it only as time management factor alone, because when I procrastinate, when I couldn't post the videos, actually it is due to my health issue. After watching those videos, even I felt like that I'm procrastinating. I've become lazy. Once a stopper, how can I become lazy? That's not possible. Okay, then I started to frame a checklist. That is, why am I procrastinating? What other than my health issues? Because of fear. It acts a threat to my subconscious mind. Okay, so students, you also might be feeling the same. That is a fear about facing the NEET exam, because obviously your brain records everything. It has got more affinity to negative thing than the positive thing. You have seen people failing and you think like you will also. So that leads to indirect procrastination. What I expect you to do is write a checklist of scrutinizing yourself. Scrutinize yourself. Okay, scrutinize yourself. What needs to be eliminated? If you have fear towards the exam, difficulty in physics, or writing down chemistry, like this, you need to sort out and you will automatically sit down to study. This procrastination will be eliminated on its own. So today I am winding up the class right now. We'll share more interesting topics both in lessons as well as the mental health, because many of the teachers, even the teachers, school, the students doesn't know the importance of mental health in preparing for exam, not only for preparing, even to lead a daily life. Okay, so if you are able to improve your focus and concentration, then you can succeed anything, not only this NEET exam. You can succeed in anything. I hope you enjoyed the session. We'll have a revision, total revision tomorrow, students, from day 1 to day 6. Tomorrow I'll have a complete revision of what we did all these 6 days. So even those who missed can join tomorrow. So now what we saw is the organelle, nucleolus. Major function is site of rRNA synthesis. The primary product is it prepares the ribosome subunits. Next, Golgi apparatus, which helps in packaging and modification of materials. The primary product is glycoproteins and glycolipids. Next, you have ribosomes. The major function of ribosome is protein synthesis. Primary product is polypeptide chain. Next, rough endoplasmic reticulum, which helps in protein synthesis and secretion, secretory proteins. Next is smooth endoplasmic reticulum, lipid or steroid hormone synthesis, lipids and detoxification. Next, lysosomes, they are involved in intracellular digestion. That is, the primary product is waste or biomolecule breakdown. And your what the mental hack yesterday I shared is about repetition. Today it's about procrastination. Don't fix yourself as a lazy one or a procrastination student. Okay, that is not the real thing. Don't listen too much of negativities. Okay, listen to a video of procrastination, and but don't apply on yourself. Okay, don't apply on yourself. Just scrutinize what is making you not to do. Okay, and work upon that each and every single day. Becoming a doctor, a surgeon, it's a great feeling. So for that we have to work upon ourselves. Okay. Okay, students, we'll meet tomorrow. Please leave your valuable comments in the comment section, and have great day. Thank you for listening and following. I'll be posting the same as shorts so that you can revise it in 5 minutes. Okay.